Everyone. Um, Welcome to Skype a Scientist Live. My name is Sarah McAnulty. I run the Skype a Scientist program. And today is DNA Day, not just for us, it's DNA Day for everybody. Today we're celebrating the Human Genome Project effectively being completed. And also just the study of DNA and all we can learn from it. Today, we are super lucky to be joined by Dr. Kathleen Gillespie, who is a DNA scientist, and will be telling us a little bit about what the deal is with DNA, why we study it, and then answer all of your questions. So thank you so much for being here with us today, Dr. Gillespie.
Yeah, it's a pleasure, Sarah. I mean, this has been really fun. Hello out there in video world. My name is Dr. Kathleen Gillespie. I go by that for now that I'm a SUNY professor at SUNY Cobleskill in upstate New York. I also go by Dr. Kate Gillespie, which is actually my nom de plume for when I'm doing my creative work, which I write and publish scientific poetry. And you can catch me at my website, The Poetry in Science. Sorry, I'll always get a plug, right?
You always should.
What am I doing here? Well, as Sarah said, I am a DNA scientist. I recently was working in Marine Biotechnology, which all sounds fun. Everybody thinks you're swimming with the dolphins and the turtles, but actually you're catching them, grinding them up, and extracting their DNA, RNA, and protein. That's the reality. So I was using zebrafish as a model system to look at genes that make more protein. It's like my gene was called EIF4E. I know that's a mouthful. We do the alphabet with these things, but what you should know about it, it's sort of like EIF4E. If we are a car, then EIF4E is a spark plug. So without that, nothing gets running. You make no proteins. Kind of important. And fish seem to have a bunch of them. The fish are weird. That's another presentation I give. Hashtag fish are weird. They basically have multiple. We may have 4, but they have 6 to 8. So I started studying that DNA that controls those genes that have all these different genes. For whatever reason, that's going to get the translation and get proteins started. What am I doing here? You're probably wondering. Well, I'm still continuing along the marine line because there's aquaculture and there's trout. Trout is big in upstate New York, because you could probably realize it's recreational, people fish it and eat it. And so we're doing a couple of different studies with it. One of them is sort of to help aquaculture. And the other one is sort of like more of just a what if we can do this kind of project. So I'll tell you about the one that's the what if is because I'm getting funded for that. The what if is actually, trout. Trout are great, but there's other fish out there that are invasive. Probably heard about that. Invasive species, they come in, they do damage. So we don't want them here, but what can we do? One thing you can do is make them so they cannot make more of each other. And that's a sterility, and there's a trick to doing it. It's actually changing the number of chromosomes they have. They're usually 2, like us, we're 2 diploid. And instead we change them to 3, triploid. So you make them triploid now, they can't reproduce. They're also kind of bigger, a little fatter. You know, that's part of like a side effect, like what we call a phenotype is looking. And there's ways you can do it. And so what we were trying to do is like, well, it takes about a year for you to figure out if you made a fish a triploid, but how about we do it like a few weeks by looking at a really earlier developmental stage and doing it by genetic means, instead of doing it by like karyotyping. And guess what? Works. So we did a proof of concept of that to say we actually get that to work, and we can look at it within a few weeks of actually them being fertilized and seeing whether or not they're triploid. And so that was fun. Yeah. And then the second one we're doing is that, of course, disease. It's disease, like I said, if you can get 6 degrees of separation to anything medical, people are going to want to know, you know, so fish disease, gills, they have to breathe, right? But what happens is that some tanks, because these are all in tanks, all these fish, they get so many of them, it's easy for them to pass around the disease, like what's called bacterial gill disease, or BGD for short. And it's a particular consortium of bacteria, and we studied 3 of them, Pseudomonas, Labobacterium, and Aromonas, to see if we can actually identify them in a tank before the tank just basically goes literally belly up. It can take almost 24 hours, and it's like from the time they know it's infected to the time the tank is die off. So we can do it a little bit quicker than that using genetic means, using something specifically called PCR, a polymerase chain reaction. Ask me anything you want about that. I've been doing that for a while. And we basically can see it within like a couple of hours, see if there is one of those particular causative agents in there. And that's also been successful. We're moving on with that. And the last one is boring because it's just bacteria that glow. Nobody cares about that, right? So the bioluminescence is actually what it is. I always say that, but check out my site because there's much better pictures there. It's kind of hard here because one, we're in the light and we usually look at this stuff in the dark. But if you ever see those pictures where you have those glowing like fish or shrimp or squid, and we use squid. We isolated bacteria that was bioluminescent. I actually wanted to know more about the DNA that actually causes it, allows it to become bioluminescent, and the genes that's in that DNA. And so we're working on that more because I'd like to actually use potentially the bioluminescent gene and use it for something else, like maybe tie it to something there's a contaminant. And when it's a contaminant there, maybe it'll glow. But that's all that's downstream, as we call it. That's something we're working on. So that's kind of a good little overview of what I'm doing. We actually just came back from a big conference for SUNY called CERC, or SUNY University conference, research conference for students. So, you know, they're having another little conference on campus. So I don't have their posters, I'm sorry, they're being sent somewhere else. But it'll be something that I'm going to share like on like probably a website, or actually SUNY will share it. SUNY Cobleskill will share it. So that's a little bit of background on what I'm doing.
That's awesome. Super cool stuff. All right, we already have a whole bunch of questions here. The first one is from Gavin. What are you studying for translational factor proteins in zebrafish?
Okay, is his question on also the chat, or you're just going to give them to me?
I'm just going to give them to you. I'm going to read through the ones in the Q&A. And for folks who are having trouble hearing, I'm really sorry. I am trying to figure out how we can add captions in addition to K for those who don't understand ASL. I'm working on that.
I have like a little bit of an experiment I wanted to like show you. Maybe I could do that as an outro. But
let's see if that works.
I'll put this on and see if this helps a little better.
Okay.
Hey, can you hear me? Is that a little
better? It sounds
better?
about the same as before.
Okay. I was hoping it would be just a little better.
Yeah, it sounds about
the
the
same.
same.
I'm talking through this, but nobody's around.
If no one's there, yeah, well, it's totally up to you. I don't want to pressure you to do anything that you don't want to do.
So, okay. All right. This might be better.
That does sound better, actually. I regret to admit it sounds better without the mask. Feel free to put it back on if people show up. But yeah, okay. So
okay.
Question from Gavin. Yes.
Yeah. So what did he want to know specifically? I mean, maybe I didn't catch the full question.
The question was, what are you studying for translational factor protein in zebrafish?
Okay, what I did study, because this was actually my doctoral thesis where I got my PhD, and I was studying what the characteristics was. Like people didn't know where we could fit them, because there's actually two things that proteins do. They either do a job or they regulate something else. And ours either initiated, and we had 3 classes. Class 1 initiated, class 2 and 3 regulated, but under certain conditions. And class 3 was more known because it's for like tumor suppression, like in like prostate cancer and all of that. You know, again, like the sexy health thing. But 2 was kind of more of a mystery, and I found something that's peculiar in it, because it turns out there's two proteins, 2 2A and 2B, and 2B seems to work a little different than what's out in the literature from the zebrafish. So that was something that was a line I pursued to find out that it could actually act as a initiator under certain conditions, which was not thought of. So it's like my little like, yay, I found that out moment.
That's
So,
awesome. That's always
yeah.
a good feeling, for sure. The next question comes in from Elena. Could you explain what you mean by triploid?
Okay, so here's how it is. Like I'm gonna look my students because, so what happens when I'm gonna say we, it's like the royal we, when there's a reproduction event, you have the male and you have the female, right? So the male has one set, of the chromosomes, because we're talking about making, something new, right? Your offspring. But the female has two. And one of these actually has to be kind of, I'm going to say, has to go away. And it's what's called a polar body, and it usually goes outside, it kind of like pushes it out. And now there's only one, so now we got the mama, and we got the daddy, and now we got two. So in making a triploid, like for the fish, there's something that has to keep that polar body in. So what is that? Typically, it's two different things. It's either heat, which is like a higher heat. These fish are kind of cold water fish, freshwater fish. So something that's more like 30 degrees centigrade. And by the way, I only do temperatures in centigrade in my mind because, yeah, science. But 30 degrees centigrade, which is like pretty hot. We're at 37. We're 37 degrees, for example. Or better, pressure. And we're not talking about a little pressure. We're talking about 9,500 pounds per square inch. That's like almost what, 2 tons, well, like 2 tons, remember what a ton is, right? Like 2,000 pounds to a ton. So almost like 10 tons to like 5 tons worth. And yes, for a short period of time, for like about a minute. And that does the job, it seems like a lot better than the heat does, which can be like variable. And there were some treatments we did, and originally when we were first doing these experiments, but the experiments are ongoing. That's a good question.
Very cool. All right, our next question. Do you use, this is from Gavin as well, do you use any chemistry in biotech or within your job?
All the time. I mean, I'm using sodium chloride. That's chemistry as part of doing extraction. We make buffers all the time. In fact, that's one thing I'm really teaching my students, how to make buffers, because you need them. TE. Yeah. You say TE to a biotechnologist, it's like saying Kool-Aid.
Yeah.
Right? It's pretty common. We have to use chemicals to do things like extract or lyse, like cells. We use chemicals for doing basically labeling the DNA. There's this particularly really good one called ophidium bromide, but it's so good, it's also really kind of toxic. It's a carcinogen, a mutagen. We use it, but we know how to handle it. We have something else that's called like kind of a DNA safe, like gel green one that we use, which is less toxic, but its job is to label, fluorescently label DNA so we're able to see it. That's the visual.
Very cool. All right. This is from Mrs. Tanner's class, from Natalie. This is a 3rd grader asking this question. Does everything have DNA?
Anything that's alive.
Sounds good.
No.
Rocks? No. Unless something died and is on top of them, then maybe you can get some DNA off the surface, but rocks themselves.
No. Rocks, no. But everything that's alive on this planet, yep, it's got DNA.
Awesome. The next question is from Noah. Is your job really cool?
Yes. Next question.
Confirmed. All right.
Confirmed.
The next question's from Christian Barkley. What are the major or minor differences between human DNA and animal DNA?
Okay. So how can I put this correctly? Because we talk about percentages a lot. Like, how closely related are we to whatever we're related to when we're doing what's called a phylogenetic tree? So how this works is that DNA is DNA is DNA. Everything has the major building blocks are 4, making sure my fingers are right. A, T, C, and G. Everything living on this planet has that. Now, how it's rearranged and how it makes genes is kind of the fun part. But like, let's just put this way. So that's just like asking me, like, humans, for example, have insulin. If I say insulin, you probably know what that. But does a dog have insulin? Yes. Do cats have insulin? Yes. Cows, and they all have insulin, but it's the insulin that has been developed within their particular species. We can take all of those insulins together and take their sequence of the ATCGs, line them up, or better yet, the proteins, and see how they look similar. And they're very, very similar to them because they all have to act like insulin.
Right.
So in a way, and then of course, they said, well, what percentage? Well, actually, I found out that I have 9%, I am human. But then that 0.1% is me, this human that, has like insulin, that doesn't have this, or has this curly hair or whatever. That's special to me. But I'm basically human. I'm 99.9% human. So we think about that when we're comparing things, and we do it in the scientific, we usually compare things boring like protein. Go at any paper, it's like, oh, look at this protein I'm looking at, and it's similar to, fish, and to cows, and to, giraffes, whatever, But it's all DNA. We all have like the same building blocks, and that's what we usually call it, the building blocks, our blueprint that makes what we need to make.
Awesome. Thank you. The next question is from Dean. Can you break your DNA?
All the time. And it's bad when that happens. When we break our DNA, when something happens, like an environmental assault, like say a chemical that you breathe in from secondhand smoke, or like when I got some, ophidium bromide on me, which, no, I don't want that. But say if I did, then okay, now it's labeling my DNA, there's going to be a problem. And what happens when DNA replicates, everything has to be the same. It has to be identical. But then you get these conditions, you get these situations, and here's a more popular one. So you like to go to the beach, probably. I mean, I'm from the shore. I'm from Jersey, so I go to the shore. But yeah, gotta remember to wear your sunscreen because the UV has the effect of taking two of those bases called thymines or T's, and it connects them together, and it makes what's called a thymine dimer. And now how's your DNA? It's like, okay, I'm trying to replicate, going along, oops, it stumbles. What was that, And now it's not the right sequence. The sequence has been made, but there's something kind of wrong with it. And those kind of things can snowball. So what's great with us is that we have what's called DNA repair systems, which is sort of like an overall like blanket term for all of these different like situations where we get proofreading, we get things that excise, that like that thymine kinase comes used to get out. Things that actually repair if there's a double-stranded break, like both of our strands of DNA. You might have just like something one, but if you have both of them broken, that can lead to major effects. So yeah. We, as organisms that do have DNA, also have ways to repair it, and some of those repair systems work well, and some don't. I mean, I have a neighbor, I'd always say this. She started smoking like a pack a day or so when she was 16. She just stopped 60 years later, okay? Her husband smoked and died from lung cancer. Her cat, who did not smoke, but had secondhand smoke, died from lung cancer. She's fine. She's literally fine, and this is like she's been smoking for more than half a century. So I tell her, I would love to get your genome. I want to see, what genes are being expressed in you. Which of course I kind of scared her a little. Like, yeah. What? But I totally want it, because whatever was working for her, it worked. And trust
either
me,
her DNA repair system or her tumor suppression.
Her, that special 0.1% that makes her her has got something special sauce in there. And I would love to know more about it, but she's older, so these things kind of like scare her a little.
Yeah.
But maybe she'll will me. some DNA,
You never know. Yeah, that's pretty cool. We've got a question here from Elena. Do you have any experience or can you speak to the use of eDNA in your work?
Yeah, environmental DNA, I'm trying to get to that. Thank you for mentioning it because I want to write a grant where I can work with eDNA that's being used from one of my colleagues that are looking at like river water quality. And then also I have now somebody who's looking for fish, a particular rare fish that's, well, let's just say they haven't seen it for 50 years and they're trying to identify if it's still there. But yeah, eDNA stands for environmental DNA. And I've been in environmental like sciences for both of my degrees. And I've done like a lot of soil science and some like bioremediation. I've done a lot of different things, nanotech. I've done a lot of different things, but I really would like to get to it. But the problem is that doing this, getting that information is costly because you have to go through special instrumentation, which is usually not in-house, like at a university or smaller one. So, but yeah, I'm totally on board, and I'm trying to write a grant so I can work with other people. Like we could probably get some money towards not getting the samples, but processing the samples, running them.
Yeah.
So, yeah.
Sounds pretty cool. The next question is from Caitlin Livingstone. What's the hardest part about starting a career in science?
Deciding you want to do it, deciding that that's what you really want to do with your life. But I'm cool with it because my life, when I'm doing research, is discovery. And things go right and things go wrong. And either way, I'm enjoying it because I'm learning something. So most people, I think, like me, I was like, oh, I want to be a vet because I want to, save a little birdie or something. And then I started reading some other books like, no, I want to look at the genes. I want to be, a geneticist or something because I'm kind of been a nerd from way back. Mm-hmm. Because I wanted to see how to make things work, not just the surface, like the health, which is great, but I just wanted to see what makes things tick. So,
yeah. Awesome. Yeah, it's really fun getting to play around and learn new things that nobody else knows. It's just fun. It's a good time. So we have a number of classrooms who got in the mail a strawberry DNA extraction kit. And one of our classrooms is asking, like, can you extract DNA for example, from a strawberry? Like, how do you get DNA out of a thing? When you start with a strawberry and end with DNA, how does that even work?
I'm actually going to demonstrate this.
Oh, good. Let's do it.
Time. Okay, gather around. Okay, I'm going to put, so mind you, this is everything that you can get from a supermarket. So I'm wearing gloves because I'm a glove wearer. I'm going to be wearing safety glasses because I'm a safety glass wearer. But we have, and you probably think we got salt, we have soap, and we have these guys.
Strawberries.
So first things first. So you have this. I'm going to be having on board our license buffer that we have water, salt, and soap. And soap is the detergent. But first things first. There is lots of DNA in these little strawberries. They basically, instead of having a diploid state like us for 2, they got an octoid with like 8. So they had 8 copies of their chromosomes. So they're full of DNA that we can literally physically see. So they're great. And here's the fun part. You put them in a Ziploc bag, and then we need to basically crack open the cells so we can get the DNA out. What's the best way to crack open things? Well,
squish them.
So I do what we call, this is the mechanical, because I'm using my hands as mechanical. I have a special equipment. We do this for tissues like in fish, and it basically makes like a little fish like smoothie, fish tissue smoothie. But this is also good. So I do that first. And then what I'm going to do is I'm going to make the buffer. So I have my water. I actually prepared things. I'm going to pour some of this in. So here goes first the detergent. Now, mind you, we're doing this is not exact numbers, but what's happening here is about 180 milliliters, about 3/4 a cup of water. This is about 20 mils of that soap, but I actually add a little extra because I know it's going to be a little on the while, and that's like about 4 teaspoons. And then I have the salt. which I already weighed out, which is like 1 teaspoon, which is like about 5 grams. So this is now our solution. And I'm just gonna do a little stir. You want the salt to go into solution, meaning so you don't actually see the salt grains anymore. But one great thing about water being the universal solvent is that salt goes into it, and now it got thick, and it's a little bit you know, frothy because of the fact that we have the soap in there. So I'm gonna take, and now I'm gonna be a little exactly, but I'm gonna put, and it's gonna be a little messy, about 20 mils. So now I'm going to use this buffer to help break open the cells. And what happens is the salt plays a role, the detergent plays a role, all the other junk that we don't want, all the lipids and proteins and everything else, we want that basically out of the way. And that's what this like buffer helps do. And of course I help it along too, because now as you see, nice, right? I use a couple extra. Sometimes if you want to do this, get these to really squish up quick, you put them in the freezer. There's lots of little tricks. And there's so many different flavors of this particular experiment out there. So go forth and discover too for yourself. There's also one video where a guy actually, after this, he dries it down and tastes it. I do not recommend doing that. But he did say it was salty, slimy, and gross. His quote words. So now we have a solution. We have our strawberry smoothie we'll not drink, obviously, because it's got soap and salt in it. No. But we also need to get rid of the big parts. So this is the getting rid of the big parts thing. So what I'm going to do is I have a little strainer here, and I'm hoping, and I'm going to collect some of this liquid, and hopefully the little big parts will just be around, but just enough to show. This is just a show. Okay. So, and I have something here for the waste. You can also do this with like a strainer. Actually, this is just a paper towel in here. And here is, oh, that's great. So what I'm going to do now, just to put this over here, I have isopropyl alcohol, isopropyl 2, which also known commonly as rubbing alcohol, and I have it in the freezer. So you're going to have to give me 1 minute or less to go and grab it out of the freezer, because it's best when it's cold to do this. It actually aids in what we call precipitation. So I'll be right back.
You guys have had really good questions today. eDNA is so cool. And everybody asking about whether butterflies have insulin, I'm working on it. I'm trying to figure that out. I don't know if they do. I don't know if a butterfly has a pancreas even.
Here I'm back. Okay. So, and what we're going to do too is like I have something to get this out, because what's going to happen when I pour this in, and I'm just going to pour in like a few mils, just enough to cover the top of it. And then I have a little now swab stick that's going to actually aid us in seeing this. So just got this fresh for science purposes. So on, and so I'm going to dispense some of that using this spout here. Goes, voila. Okay, I'm going to show this. I'm going to bring it as close as possible. You probably see some slimy stuff at the bottom, right on the top of this. I know there's a little bit of that slimy stuff is DNA. And with strawberries, you often get a lot of it, enough where you can take a little stick like this and just sort of draw some up, get the nasty, it's kind of snotty. And you can take, it's a little sticky, it's kind of harder to see from here, and I'm holding it over my computer. I'm like, oh, I don't want isopropyl on my computer. But this layer here is that DNA that was in that solution from the strawberries, now becoming partitioned into the Okay, and then I'm like, whoa, look, there we go. See, it's not, see it?
Blobs. Cool.
And by the way, I've done experiments where I was like, well, what about bananas? Not so much, not just because of the amount of chromosomes, but because bananas smash differently. They don't come into solution as well. I don't know.
Yeah.
Try blueberries, not blueberries, berries, like blueberries, blackberries, they kind of work. So, you know, it's not just a strawberry, but the strawberry is like the one you can usually get the best bang for your buck as far as DNA. The ooh factor. So when the kids see it, after I let little kids, like, you're going to be these scientists and you're going to set this up. And they see the fruits of their labor, like, oh, look. And people sometimes save it in like a test tube. Some people will put it into a little necklace or something. I'm good. DNA. But yeah, so that's like the quick, and see it when you have everything together, it takes no time, and yet it's science. We use the same principles in extracting DNA from fish, extracting DNA from bacteria. It's all the same. Yeah.
Yeah. What I didn't realize before I became a lab scientist is that sometimes you're using the same basic techniques on all sorts of different life forms, but you never know what's going to be a problem. Like if you had never played with that banana, you might not know that bananas are just a total pain in the butt to get DNA out of. And sometimes when we're working with like goopy bacteria, like some bacteria are pretty ungoopy, and other bacteria in squid that I used to work with are super goopy, and getting DNA out of the goopy stuff is just like so hard. Yeah. And you never know what those challenges are going to be until you start playing around and learning stuff.
I have a challenge, and like talking about the triploid again, going back to that, we first tried it with when it was first fertilized, and it's in like the embryo. It's just basically a little ball of like sac, which is like this fatty lipid material called vitellogenin. And let me tell you, it's hard to try to isolate DNA. It's hard.
Yeah.
So we kind of went, let's go to when it's meaty. When it has a little, when it literally looks like a tiny baby fish with a sac of that, like, because it's still using that. Mm-hmm. But now it's getting, so it's easier to extract.
Sounds good. We've got a question from Christy B. Can you tell us a little bit more about heterozygous and homozygous genes?
Wow, I feel like somebody's got an exam coming up. Okay, so homozygous, and I guess in reference, like you talk about with genetics. So the homozygous is homo being the same and hetero being different. And like I can just use like the classic example, like when you have classic being Mendel, classical genetics. So I teach a monogenetics, which is like using mono techniques. But let's go back to Mendel, and he would have something that would be with a pea plant, like a yellow color or a green color. And the green was like homozygous. You have green, yellow, and one was, and this is something now I gotta have to tie it in. It's like one can be dominant, which is expressed, and one's recessive, right? So if it's both the same and they're dominant, then the dominant will dominate. Okay, and if they're both recessive, then it will be recessive. And when you start crossing them, you find out putting two dominants together or putting one dominant and one recessive together, that's where you get your heterozygous. Sounds and there's good. Like lots of classical experiment. I mean, I don't know, I didn't think I would be doing Punnett squares here, but you know.
Yeah, yeah.
So
we don't have to get into Punnett squares, really just that every gene that we have, anyway, has two copies. And if it's homozygous, you got two of the same thing. And if it's heterozygous, you got different things. Yeah. And that's how that all works. The next question is from Gavin. How exactly do you sequence a genome? How does that even work?
Ooh, well, thanks to computers, because it's a lot of compute power.
Yeah.
The kind of sequencing is going on now, we call next generation sequencing, though technically we're in third generation. Mm-hmm. It's like Pacific and everything. So how does that work? Well, it takes something that was originated by a scientist called Sanger, sequencing, mm-hmm. where he was able to find the exact like allocation sequence being, there's an A, there's a T, there's another T, there's an A, there's a G, there's another G, and all of that. He was able to do that by using a chemical reaction that he developed where it was either would add it to, I don't know how much detail I should get into it, but either you got to add it, oh, no, this one, it stops. And the one it stops at, now that's the one, that's the next in line. So it sort of worked really great for like a sequence at a time. But when we talk about next generation sequence, we're talking about massive, what they call also massive parallel, we're talking hundreds of thousands to millions. So the data that we have out there now for genomes, like our genome, that's, I guess I ask my students, like, so how big is the human genome in base pairs? And somebody said 1 trillion. And I was like, well, this is The Price is Right. And you're a little over, but you're in the ballpark because it's just like 3.2 or 6, well, you know, it's changed again, billion. And we just actually totally, truly closed the genome this year. It's truly closed, meaning we do know the human genome. There were some things that were a little missing from 2003, and they've been working on it for, I guess, almost 20 years now.
Yeah.
But there we go, you know, science marches on. And so now we know completely the human genome, and they found some new genes and stuff. I have an article that just came out in Nature recently about that. So that's real cool. But awesome. Yeah, doing the sequencing takes, and again, without having computers being able to sequence things back even in like the early 2000s, they would not have been able to do it. Yeah. It's a really great, and I would totally, if you know what TED Talks are, you can search for one on YouTube, a TED Talk that talks about like the race for the DNA, because it was kind of a race for understanding the human genome. And the two big players was Francis Collins at NIH, and of course, Craig Venter, the dude who actually figured out like the type of method that we still use today as far as like doing other genomes, like whole genome shotgun. Yeah.
Very cool. We've got a question here from Dean. Can animals still have DNA after they've turned into a skeleton?
Actually, you can extract DNA from bone.
Cool.
And teeth. And teeth. Someone was asking about that. It's possible. And in fact, I don't know, anybody remember Bones, like the show?
Yeah.
He did that all the time.
Yeah.
And yes, you can. I mean, because there is still the signature of DNA. In fact, I read up that DNA has a half-life about 580 years or so. So yeah, if a person has been around, so half-life meaning like you'll get a diminishing amount available. But it also says that no, we can't have Jurassic Park because it's too long. It's too long ago. So you have to have a time machine. Then, like, you know,
and then you can do it. Sounds good. Our next question is, what was your most favorite and least favorite class from when you were in high school?
Okay, my most favorite class was biology. Okay. Oh well, actually, it's dual biology and English, because I'm writing. Remember, I told I'm a creative, so I was writing a lot, and I really liked writing, like, you know, poetry and weird little stuff. So biology was like, oh, like, this is cool. But I guess I will have to say that chemistry wasn't really my favorite, but don't blame the chemistry, blame the professor, because she did not make it fun. And I was also told things that are not exactly politically correct at the time. And I will say that. I'm sorry, I have to say that. But yeah, not my funnest class, but you know, I did it, I got through it. You know, I was, and same thing comes from, you know, college, you know. Microbiology though is my fave. That is like me. I'm a micro person, micro gal.
Very cool. Yeah, I also really liked English class, and I ended up being a scientist too. So
yeah.
You know, whatever. So, okay. The next question is cool. Do you ever make mistakes? Do scientists make mistakes? And how do you fix mistakes when you make them?
Okay. So I actually want to create a course. It's called Lab Failures 101, because my students, and I'm saying my students, the ones that are doing my research and the ones that are taking my class, when things go wrong, they get like, what are they going to do? And I'm like, well, in research, things go wrong. we're discovering things, so we don't know. We're like shining flashlights into the dark. So what do you do? So I have 5 students that every time they make a mistake, and they do make mistakes, I said, I am not grading you on your mistake. I'm grading you on what you do afterwards, how you go forward. So the gel didn't run. Okay, so what happened to the gel? Was it something in the media? You know, was it the sample? What was it? Like, kind of you make the decision and how we're going to go forward. So yes, mistakes are what keep people. Some people don't like making mistakes. They're perfectionists. Well, you know, science is also sometimes an art, and art can be messy. But we learn a lot from our mistakes in science, and everything we learn from these guys doing, they've learned and have gone on to do something else. And you also get your eureka moments, like, you know, hey, that seems a little weird, but I know I did the right science, but it should be this. So let me go and do something else and check it out, you know? So yeah.
Very cool. Sorry, my cat was like really needed a treat immediately. So I had to mute for a second for him. Okay. So at this point, we are almost out of time. We've been talking about DNA for almost 45 minutes. And we like to ask everybody the same 2 questions at the end of our sessions. The first question is, if you had everyone's attention in the whole world and you could tell them one thing related to your area of expertise, what would that be?
Fish have an extra 4E that's not found in any other, you know, eukaryotic system, and it's called 1C. It works better than 1A because that's where we have the 1A is like in everything. Why? What happens if we put a 1C into something that lives on land? I'm very curious. That was my question in my dissertation. I'm like, not that ethically I'd do it,
right?
But I am curious. You know?
Yeah, that's cool. All right. And then our second question is, you still have everybody's attention in the world. You can tell them one thing about anything. It could be about your area of expertise, or it can be about something completely different. What do you tell them?
So this is a worldwide audience. I tell them to like sit. Science is real. Science is, you know, fact. Science, please, you know, believe the science, because us scientists work hard to make sure that all the science is things that can help not just humanity, but the world, you know, itself.
Yeah.
So please listen. Please ask questions like you're doing.
Yes.
You know, and keep doing it.
That's right. Did you want to show us a gel before we say goodbye?
Yeah. Okay, I'm going to take you on the road a little bit here.
All right.
Okay, here we are. So testing of VLC is like where you do stuff like culturing. You know, it's a safe zone. It's still there in the air. And this is a PCR machine. Yeah, is an imager of you. But I have something you can do this. I can use it.
So if you couldn't hear, the first thing that Dr. Gillespie showed us was a hood. And that's where if you want to keep the things in that area safe, or you safe from those things, you put them in there. And then the second thing was a PCR machine. And the PCR machine is where you do this specific type of experiment where basically it's changing the temperature in cycles that allows the experiment to work. So you always have to put your samples in that machine in order to, and now we can hear you great. Great.
And now actually you could also use something like this, which is a mini PCR. Ooh, and this is what we've been using, you know, in the classroom and for doing my research. And you can see I can fit it in my hand. It's got 8 stations. It's a PCR machine.
Cool.
Yeah. And this is their little gel box, and it actually has a little dark one here. Unfortunately, I can't show you. It's green because it's like gel green. It's like stains the DNA green. But I can show you what my student took on this imager, which is the BioRad one, which is like a fancy. That's fancy. Because you want to do the fancy one if you wanted to put it in a paper.
That's right.
So,
That's right.
so again, I'm going to have to like, you meet this and this meets this. So you can see here, and I'm going to tilt it a little forward. That is a gel. You can see the black bands. Those black bands, those represent DNA. The one on the far hand side that looks like a ladder, we call it a ladder because it's different sizes of DNA that's known. Commercial manufacturers make these and sell them to scientists, and that way we can determine how big in base pairs our DNA is. So she has a gel here and she's run a few samples. I don't know, because we're talking about it. She ran a gel from before and I'm like, hey, I'm going to show you the gel, but I can't tell you what sample except for they are bacterial. I know, and they're probably from the Aeromonas because she was going to work on that.
Cool.
So that shows that, and it's from samples from tanks in the aquaculture to show that yes, we can find those 3 species on tanks in aquaculture, which could be a problem. Because that could be where they're coming from. That could be the reservoir. So, so meet some DNA on gel. And
Very
she's going to
cool.
clean it up and she's going to put it into, yep. All righty.
Awesome.
Yeah.
Well, thank you so much. This was so
Okay.
cool. Is there anything else you'd like to plug before we say goodbye?
Let's see. I plugged my thing. I mean, I'm doing actually a workshop in Brooklyn in July for that. And I do actually do workshops if you're interested in having me help people, from young to old, scientists to science enthusiasts, like learn about science through poetry. I am available. You can check out my website, or you can also contact me, here through SUNY Cobleskill.
Awesome. And I've put the link to your website in the chat at the beginning and now at the end.
Okay.
Thank you so much for sharing all this with us. This was super cool.
Okay. Yeah. Thanks, everybody.
Awesome. Thanks. And thank you, Kay, for interpreting. This is Kay's last day with us after interpreting so many sessions. So thank you, Kay, for being a part of our program.
Yeah.
And helping us understand what's going on. Everybody, thanks for coming. We will see you at our next session. You can always see what's going on at skypeascientist.com and then click events and Skype a Scientist Live. We'll see you at the next one. Thank you all for coming.
Thanks, everybody. Back to science for me.
Awesome. Thanks. Bye-bye.
Okay. Bye.